So we have Natalie Herbison presenting on her work Body Size Nor Sex Influence Critical Thermal Limits in a Solitary Spring Bee.
I am Natalie Herbison. You do pronounce the H, unfortunately. I am an undergraduate researcher from the University of Kansas, and this study was the accumulation of my findings from my undergraduate research project. Uh, I did this on Coides inequalis, and the study is body size nor sex influence critical thermal limits in a solitary spring bee. All right, so who is Coides inequalis? So Coides inequalis is one of our native solitary ground nesting bees in North America. The really nice thing about Coides inequalis is that it comes out pretty early on in the year. So we see males start to emerge in about mid-March, and then we get females 1 to 2 weeks later in late March, early April. And we'll have females all the way through early to mid-May, which in Kansas, which is where we did this, we conducted this on the University of Kansas main campus. Uh, we have major temperature fluctuations throughout the day. So we have a lot of very different uh, just thermal exposures and phenologies on our campus that might be slightly different from things that you would see elsewhere, just because we do have those major fluctuations seasonally and daily. So we set out to kind of determine if males are more thermal tolerant than females because of that early emergence time, or if the body size of the bees would influence their heat and cold tolerance. We kind of looked at this through deciding that males would display greater cold and heat tolerances than females because they're exposed to greater fluctuations in temperature. Kind of going back to that daily fluctuation, we see temperatures in early spring as low as 30, 40 degrees, all the way up through 80 degrees, which kind of varies from day to day. And then we also hypothesized that larger individuals would display greater cold and heat tolerance. This is kind of based on previous studies that showed that larger individuals heat up a little bit slower, but because of that larger body size, they're able to kind of accommodate for a wider range of temperatures and thermoregulate accordingly. So why are we doing this? A majority of the studies that are out so far on thermal tolerance are on bumblebees. There's not a whole lot on solitary bees, and it's pretty much exclusively tied to bumblebees. So what we really wanted to do was look at this solitary bee and see how it would respond physiologically to changes in temperature. We also wanted to kind of analyze if there was going to be an influence of the sex of the bee on the thermal tolerance, if males or females would have greater thermal tolerance than the other. And then we decided that this would be super important because morphologically, these characteristics might better prepare bees for variable regional and global climatic conditions that are associated with climate-related change. So in Kansas, because we have those super large daily and seasonal temperature fluctuations, we kind of figured that this would be an ideal species to test that on, because Calididae is one of the more common families, and then also just because this specific bee species is especially abundant on our campus. It is the first bee out in the spring aside from honeybees. So we decided to use them. And what we did with this was we collected about 70 to 80 male and female Coides inequalis from across our main campus. We utilized dynamic and static protocols to kind of determine what their full range of thermal regulation capacities was going to be. Dynamic protocol was using ALARA, which is an iotherm computer device that's seen here in this corner. Basically what this is kind of akin to an electric stove, except you have two burners right next to each other. And the two glass vials that are featured right here allow us to see what the temperature inside of a vial is that the bees are experiencing. And then we take the temperature from the two different sides of the plates and we average that out. And that is hypothetically what they are experiencing inside of the vial. So up in this top right corner here, those are some honeybees in the testing vials on the ALARA hot plate. And then for static measures, we utilize chill coma recovery. We took these and we placed them into plastic vials, capped it with cotton, similar to how we do for the ALARA protocol, except we put them into a cooler full of ice for about an hour. And then we would pull them out and let them thaw ambiently in room temperature of about 72 degrees Fahrenheit or about like 24 degrees Celsius. And we would time how long it took for them to recover from that colder exposure period. What we found from this was that there is no real difference between male and female for both CT-min and CT-max. This could just be due to the fact that both of them are exposed to that super wide range of temperatures. And then this again just shows that there really isn't a whole lot of difference there. And we also see that the body size of the bees doesn't influence their thermal tolerance either. So those were our two main hypotheses right there, and they kind of got knocked out all at once, where we didn't see any real correlation between body size or the sex on their lower critical limit, so the coldest temperature that they can be exposed to before they lose metabolic control, and the highest temperature that they can be exposed to before they lose metabolic control. Now for the chill coma recovery time, we actually did see something slightly variable. So again, we saw no correlation between the body size on the recovery time, but we did see that males had a longer recovery time than the females did. And this could potentially be due to the nutrition of the bees, because the males come out about 2 weeks earlier than the females in mid-March when there really isn't a whole lot blooming. They're kind of opportunistic on both wild and agricultural plants that are available. So we think that part of this delayed recovery time has to do more with the nutrition of the bees than their body size, as we're seeing over here on this left side. Now, this does require further exploration. We have plans to continue this coming spring when the Cleetes and Aphallis are out again. This time we hope to take into consideration urban hotspots versus rural areas. So because we collected all of our bees on the main campus of KU, which is located in the city of Lawrence, about population is probably like 200,000 maybe, just between like people coming in and out from Kansas City on a daily basis and then all the time population. We have a lot of CO2 buildup relative to surrounding agricultural lands. So we think that there is potential that increase in temperature caused by just local pollution from cars and ambient heat of buildings might be influencing the thermal temperature. That's something that we're hoping to further explore this coming spring when they're out again. And that is that.